Very good evening, children. Can you all see me? Can you hear me? Very good evening to all. Can you all see me? Can you all hear me? Balakrishnan, Charvi, Subarna Lakshmi, Madhav Krishna, Kadiravan, Deepak. Very good. Yes, children. Thank you. Thank you very much. Shall we start our class? Okay. So today's class, the topic for today's class is cell structure and function. Cell structure and function. In that, cell structure and function, we're going to talk about what is a cell and the various structures that are present in the cell. So first let me tell you, explain about what is a cell and characteristics of cell, and then we learn about the organelles and all those things. Check, is it volume is low for you all, children? Is that volume is low for you all? I think from here everything is perfect. Check, check, check. Are you all able to hear me clearly, children? Check, check. Check, check. Okay. Very good, ma. Very good. Thank you. The topic is about the cell. So what is a cell? We all know that cell is a structural and functional unit of life. What is the structural unit? The cell gives a definite structure to the any organism. The structure of organism, it depends on that cell. Similarly, the fundamental unit, it is also called as functional unit, because all the functions are carried out by the cell, carried out by the cells. Cells become tissues, tissues become organ, organ become organ system makes an individual. So here the whole individual, it starts with the molecules. The molecules forms the cells forms the tissues forms organ forms the organ system, and organ system forms the individual. So that is the reason why the cell is known as a functional unit. The main function, for example, in unicellular organisms, all the functions are carried out by the single cell. Respiration, digestion, excretion, reproduction, everything is carried out by a single cell. That is the reason why the cell is known as the functional unit. Functional unit. The smallest known cell, it is called mycoplasma, and ostrich egg, it is known as the largest egg. The largest cell, it is the ostrich egg. And this cell, what the cell it will do, cell, it converts the nutrient that is from the food into energy. That process is called respiration. The energy, the food what we get, from that food, the energy is, the cell generates the energy. Whether it is unicellular organisms or multicellular organisms, the generation of energy, it is done by the cells and the tissues. If you observe the cells are of different shapes and different sizes are there. Nerve cells, muscle cells, cardiac cells, cardiac tissues, epithelial cells. So cells of different shape and different sizes. And the cell contains the cell organelles. Those cell organelles, it carry out the vital functions that are required by the organisms. So all the cell organelles are highly specific. It carries its own specific function. The cells we can categorize into two types. One is called the somatic cells and the sex cells. The somatic cells, it controls the bodily characteristics, whereas the sex cells, that is the ovum and the sperm, these sex cells, it determines the sexual characteristics of the organism, the male or female. That is done by the sex cells. Now we are going to talk about the discovery of the cells. The discovery of the cells in the 1595, Janssen is the first person who identified the compound microscope. In the year 1655, first time, Robert Hooke identified the cell in the cork of a tree, that is the bark of a tree. In 1674, Leeuwenhoek, he identified protozoa. In the year 1833, Robert Brown, he identified that a nucleus exists in the cell in orchids. In 1835, Felix, he identified that the living juice in animal cell, that is the cell, actual cell. Why it is called as living juice? Because the cell has certain functions, and he named them as sarcode. And 1838, the well-known cell theory was proposed by Theodor Schwann and Matthias Schleiden. And 1839, Purkinje, he identified and he gave the term called protoplasm, that is the living part of the cell. That is the living part of the cell, or he named it as sap. Sap means fluid-like structure that is present in the plant cell. Protoplasm means living part of the cell that excludes the cell membrane. The cell membrane, it is not included in protoplasm. Only the cytoplasm and nucleus are included in that. In 1840, Albert von Roelker, he has identified the sperms and egg cells also exist, or the sex cells are also there. And in 1846, Hugo von Mohl, he indicated the importance of protoplasm, because people thought the whole cell is very important for organism, whereas the living part of the cell, protoplasm, only does all the functions. In 1856, Prince M, he observed how the sperm cell penetrate into the egg cell. This is what the discoveries. Other discoveries by Robert Virchow. We know about the cell theory that was proposed by Matthias Schleiden and Theodor Schwann in the year 1838. But that theory, it was added or rewritten by Rudolf Virchow. In that he concluded that omnicellulo e-cellulo. That means cells are formed from pre-existing cells. Cells are formed from pre-existing cells. Omnicellulo e-cellulo means cells are formed from pre-existing cells. 1857, Kolker, he identified mitochondria. He described the mitochondria. And 1868, Huxley, he proposed that protoplasm is the physical basis of life. 1879, Fleming, he identified the behavior of chromosome during mitosis. What is the behavior of chromosomes? The chromosome, it will be in a form of thin thread-like structure. Later, it condenses and become a tetrad structure. Do you know that cell division process? The thread-like structure, it will be there. It condenses and become the chromosomes. That he has identified. 1898, Golgi, he described the Golgi apparatus. That's why that is named as Golgi apparatus, Camellia Golgi. 1938, Behrens, he separated the nucleus from the cytoplasm by centrifugation method, centrifugation, rotation. By that method, he has separated the nuclei from the cytoplasm. And 1939, Siemens, he first commercial transmission of electron microscope was described. And 1952, Gay and co-workers, they have established that continuous human cell line, that is the tissue culture process. In 1981, the first transgenic mice are the fruit flies are produced, and also mouse embryonic stem cells were established. 1996, the first cloned sheep, the first cloned mammal sheep Dolly was produced by Dr. Ian Wilmot. In 1998, mice are cloned from somatic cells. These are all the histories. And here there is a doubt by Madhav Krishna. What is sarcode? Sarcode and protoplasm are same. So here you just observe it here. Felix, he identified the living juice that is protoplasm only. Got it? So that only is named as sarcode. So yes, what you have assumed, it is correct. Next is cell theory. Cell theory, there are few postulates of cell theory. In 1833, German botanist Matthias Schleiden and German zoologist Theodor Schwann, both of them, they did a joint research, and then they have identified that plants and animals are made up of cells. They only told that cells are the basic fundamental unit of life. Later, Rudolf Virchow, he added some more points to that. So cell theory, it was proposed earlier, it was proposed by Matthias Schleiden and Theodor Schwann, and later it was rewritten or it was added by Rudolf Virchow. So what are the main formulation of modern cell theory? All cells are made up of cells. All organisms are made up of cells. The second key point, new cells are formed by the division of pre-existing cells. That is called omnicellulo e-cellulo. That means cells are formed from pre-existing cells. The third thing, cells contain genetic material which are passed from parents to the offspring or parents to the daughter cells. The next modern cell theory talks that all metabolic reactions take place within the cells. This is the modern cell theory. Next, we are going to talk about the exception to cell theory. Exception to cell theory. There are few organisms, they are exception to cell theory. For example, the viruses are known as puzzle in biology. Viruses, viroid, prions, these are all except to cell theory. Why? The viruses till today, we cannot categorize them into living or non-living characteristics. When it is outside the body or outside the cell, it behaves like a non-living component, a crystallized form. It will be like a crystal, like how salt crystal is there. Like that, it will be there. For many years also, it can be like that in a suitable substratum. Whereas, once this virus enters into the host cell, a living cell, it behaves totally different. So that is the reason why the viruses are known as a biologist puzzles. These organisms, the viruses, viroid and prions, they lack protoplasm. Living part of the cell is not there. They always ex exhibit as an obligate parasite that lives inside the body of other living organisms. Next is we are going to talk about the characteristics of cell, or it is called the cell doctrine. Cell doctrine. What is the cell doctrine is all about? The important informations about the cell. The cell provides structure and support to the body of the organisms. For example, if you take paramecium, the paramecium structure, commonly it is known as slipper animalcule, because the structure is like a slipper. So that structure is because of the cell. Similarly, cells are organized into different organelles, but they are surrounded by separate membranes. For example, if you take a cell, inside the cell, what it will be there? Inside the cell organelles it will be there. Each and every cell organelles, there are distinct structure, but they are surrounded by membranes. Some of them they are surrounded by single membrane, some of them by double membrane. For example, the vacuoles are surrounded by the tonoplast, single membrane, whereas the mitochondria, chloroplast, all these structures are surrounded by two-layered membranes. Next is the nucleus. The nucleus, it is the master of the cells, because that is the one which carry the genetic information that has to be carried from one generation to next generation, or from parents to the offspring. And also this is the one which contains the code for reproduction, growth and development. So all those activities are controlled by the nucleus. That's why nucleus is known as the master of the cells. And this information, the hereditary information that is present in the cell during cell division, it is transferred to the daughter cells. It is transferred to the daughter cells. During cell division process, all those genetic information that are present in the chromosome, when the chromosome divides during the mitosis or amitosis process, the metaphase stage, all of them arrange at equatorial plate. And in anaphase stage, telophase and anaphase stage will get separated. During that time, what will happen? The nucleus divides and it carries information to the daughter cells. Next is the mitochondria. The mitochondria, it is known as the powerhouse of the cell, because that is the one which involved in the process of cellular respiration. Cellular respiration. It is the place where exactly the oxidation of food process takes place. What is oxidation of food? The glucose molecule, to the glucose molecule, the oxygen is added to that. Oxygen is added to that. That is called oxidation of food, and it releases carbon dioxide and energy in the form of ATP. That is the reason why the mitochondria, it is important, because that is the one which provides energy for all physiological process. Without the physiological process, the life will not sustain. Next is the endoplasmic reticulum. There are two types of endoplasmic reticulum it is present in the cell. One is called RER, ACR, the rough endoplasmic reticulum, and smooth endoplasmic reticulum. The functions of this ER, endoplasmic reticulum, it is like a skeletal system. For example, we have the skeleton system inside, that only gives a structure to the body. Similarly, the structure to the cell, it is provided by the endoplasmic reticulum. The next characteristics about the tracheids and vessels in plants. Similarly, horn cells in animals, the horn cells, all these are all remain dead cells, and they remain functional even after the death. These are all the cell doctrines. Next, we are going to talk about the structure of the cells. The cell has a typical structure. Any cell, if you take it, The cell has a plasma membrane, cytoplasm, and nucleus. Whereas in the case of plant cell, additional one more protective layer it will be there, that is called a cell wall. Whereas all the cells, they have plasma membrane, cytoplasm, and nucleus. Cytoplasm membrane is the outermost membrane, whereas the living part of the cells, that is protoplasm, it includes the cytoplasm and nucleus. Nucleus is the one which contains the chromatin threads which become chromosomes, and that is the one which carries the genetic information about that generation to the next generation. The cytoplasm, it consists of cell organelles and cell inclusions. Cell organelles and cell inclusions. The cell organelles, for example, few cell organelles are given in endoplasmic reticulum, ribosomes, lysosome, Golgi apparatus, mitochondria, centrioles, chloroplast, plastids, that centrioles. These are all examples. Cell inclusions, that is also present in cytoplasm, that is reserve food materials, gases, inorganic crystals, and excretory substances. These are all the cell inclusions. The cytoplasm contains cell organelle as well as cell inclusions in that. Children, we go for a short break now. For 5 minutes, we'll go for a break. Meanwhile, if you have a doubt, you can post your doubts now. And after 5 minutes, I'll explain to you about the next topic, that is the plasma membrane, structure of plasma membrane, various models of plasma membrane, and how plasma membrane helps in transportation of materials. All those things I'm going to explain to you. We'll go for a short break, children. Just you also go for a break for 5 minutes. Come back after that. Children, welcome back. Shall we start the class? Shall we continue? Are you all there? For Kadidavan, no doubt. Okay, others, what about others? Charvi, Balakrishna, Subarna Lakshmi. Okay. The next topic, it is about the plasma membrane. So we have know that what the cell contains. Next is about plasma membrane. Plasma membrane, this is the one that covers the cell. This is the cell. The cell is covered by a membrane. That membrane is called cell membrane or plasma membrane. It is also called as plasma lemma. Plasma lemma. This is the thin membrane that covers the cell. And what it is made up of. This is a typical structure of a plasma membrane. The plasma membrane mainly it made up of lipids, protein, and carbohydrate. Lipid, protein and carbohydrate. That is the cell membrane. The cell membrane, it delimits the cell. What is delimiting? That is the one that restricts the cell. That is the one that separates the external environment and the cell. That's why the plasma membrane, it is delimits the cell membrane, delimits the cell. And also, it protects the internal organelle, the cellular constituents that are present in the cytoplasm. That is the reason why the plasma membrane, it is a layered membrane, a phospholipid bilayer membrane. It's a phospholipid bilayer membrane which gives protection to the cellular constituent present there. And the cell membrane, it is also known as selectively permeable membrane. Selectively permeable membrane. Selectively permeable, that means it allows only selected molecules to move inside. It will not allow all the molecules to move inside. If the cell requires that, then only it will allow that. For example, continuously water will not be taken by the cells. Even though water is required by the cell is not continuously absorbing the water. Only if it is needed, if it is highly required by the cell, then only it will absorb that. So whatever molecule is there outside the cell, it will not be taken inside. This membrane, the selectively permeable membrane, plasma membrane, it protects the entry of substances in and out of the cells. It is highly bounded to the semi-fluid cellular constituents. Next is about, we're going to see about the various models of plasma membrane. This is called Singer and Nicholson model. Singer and Nicholson model. It's also called as fluid mosaic model. In the fluid mosaic model, just observe the picture here. What are all present there? So glycoprotein, that means glucose plus protein. Carbohydrate, glucose molecule. Glycolipid, glucose plus lipid. Cholesterol, it will be there. And in the bilipid layer of the plasma membrane, there are lot of protein channels are present. These proteins are helps in transportation of materials. It is an integrated channel through which molecules move inside and goes outside the cell. There are linear proteins are there. There are globular proteins are there. So you can see that there is a globular protein here. These are all linear proteins. This is a protein channel. This is an integrated protein channel that helps in transportation of materials within the cells or outside the cell. This is a typical model of Singer and Nicholson fluid mosaic model. The next model, it is called Davison and Daniel model, which is known as sandwich model. Sandwich model. The sandwich model, the bilipid layer, this is the phosphobilipid layer. This bilipid layer of plasma membrane, it lies within the globular proteins. So here there's a globular protein, here also globular protein. It's called membrane proteins are there. In between the membrane proteins only, the phospholipid bilayers are present. This is the sandwich model. This is called a sandwich model. The next is we're going to talk about how the plasma membrane allows the molecule inside or the flow of material within the plasma membrane. What are the forces that are involved in that? One is the diffusion. We all know what is diffusion. It is the movement of molecules from region of higher concentration to the region of lower concentration. Always molecules will move from region of higher concentration, region of lower concentration, or region of higher concentration to the region of defect, or source to the sink. Where higher concentration is there, somewhere lower concentration it will be there. So always molecules, it will move in higher concentration to lower concentration. So this is one force that is involved in transportation of material within the plasma membrane. The next is called facilitated diffusion. What is facilitated? Facilitator, that means a person who helps. That is called facilitator. Here the molecules, it will move within and outside the cell. If this is outside cell, this is inside cell. So molecules can move inside, molecules can move outside. So it can move inside, can move, go outside. So that is done with the help of protein channels. So you can see that this is the protein channel which is present in the cell membrane. In that cell membrane, with the help of these protein channel, the molecules move inside and it comes outside. All these are all integrated protein channel that is present in the plasma membrane. Here in the facilitated diffusion, the molecules, it will move inside the cell and outside the cell, actually by higher concentration, lower concentration, through a protein channel without using energy. That is called facilitated diffusion. It is same like diffusion process, but what is the difference here? The molecules move through the protein channels. That's called facilitated diffusions. So who is the facilitator here? The integral proteins are facilitators. Next is osmosis. What is osmosis? The movement of molecules of region of higher to lower concentration through a semi-permeable membrane. So here, this is the semi-permeable membrane through which the molecules move from higher concentration to the lower concentrations. Here, usually the movement of water molecules, it occurs mainly by a process called osmosis in the living cells. Molecules will move from higher to lower concentration, keeps on moving like that, higher to lower concentration. Once it becomes equal, that process stops. Osmosis process, it acts from higher to lower concentration. Once the concentration, it is equal in both the sides, then osmosis process, it will stop. Next is plasmolysis. So what will happen when a cell loses water? When a cell loses water, when you keep a cell in a hypertonic solutions, it loses water. When it loses water, all the cell content, it will shrunk. That process, it is called as plasmolysis. Water molecules moves out of the cell. When water molecules moves out of the cell loses its turgidity. It loses turgidity and it becomes shrunk. That process is called plasmid. And that whole process is called plasmolysis. If the plasmolyzed cell is kept in a hypotonic solution, it gains water and it will become turgid. When cell loses water, it becomes flaccid. When cell gains water, it will become turgid. That important concept you need to understand. Next is active transport. Active transport. There is not much differences between selectively permeable or semi-permeable. Both are same. Semi means of permeable. Selectively means it will only allow certain molecules to move inside. As I mentioned already, even though concentration is higher outside, whatever is required by the cell alone, it will be absorbed. That's why it is called semi-permeable. Selectively permeable means there are so many molecules, different kinds of molecules surrounds the cell. But only what is required by the cell, it will be allowed inside. That's why it is called selectively permeable membrane. But the concept, the term both indicates the same. There is no much difference in that. Got it, children? Next, we are going to talk about active transport. What is active transport? In the previous transportation, osmosis, diffusion, we know that the molecules are moving from higher to lower concentrations. Whereas in active transport, molecules move from lower to higher concentration. Lower concentration to higher concentration. When the molecules move from higher to lower concentration, it uses lot of energy. It uses lot of energy. With the help of energy, ATP molecules, the molecules, it will move against the concentration gradient. That is called active transport. Active transport. It actively transport against the concentration gradient. What is against? Lower to higher. Lower to higher with the help of ATP molecule. That is called active transport. Active transport. Next process is called endocytosis and exocytosis. The term endo means inside. Cytosis refers to the cell. When something moves inside the cell, can you all see that? From outside the cell, the molecules are moving inside the cell. That is called as endocytosis. Here, from the cell, molecules, it will move out. The unwanted molecules, it will move out. That is called as exocytosis process. Endocytosis, exocytosis. In exocytosis process and also in endocytosis process, what is involved here? You can see that cell membrane is involved in formation of vesicles. The cell membrane, what the expelling out of the material, that is called exocytosis. Unwanted material, it will be sent outside the cell. The waste material slowly what will happen? It moves and get attaches to the plasma membrane. With the help of the plasma membrane, it will be exported out or expelled out to the external surface, external cell space. So endocytosis and exocytosis process, it usually occurs in the cell with the help of plasma membrane. Next is phagocytosis. Phagocytosis, it is an important process. It is also called as digestive process. The phagocytosis process, with the help of that, some food, it will be taken inside and digested. Similarly, the pathogen, the disease causing organisms, it will be taken inside and it will be digested. The phagocytosis process, suppose this is the bacterium. The bacterium, it will be pulled inside. Once it is taken inside by the cell, it forms a structure called phagosome. With the help of this phagosome, what will happen? The lysosome that contains the digestive enzymes, it gets attaches to that. Can you all understand, children? The phagosome is formed here with the help of the plasma membrane. Along with the phagosome, the lysosome which contains the enzymes that helps in digesting the molecules or which helps in degrading the pathogen, it will join with that. Now the phagosome along with lysosome, it becomes phagolysosomes. Then what will happen? With the help of enzymes, the microorganisms, the bacteria, it gets completely digested. And then you can see that exocytosis process takes place. Here, this is called endocytosis process, and this is called as exocytosis process. This process, it is an immune response. Even in our body, the phagocytosis process takes place with the help of the white blood cells. That is immune response, killing of the pathogen, digestion. These are all the process that takes place within the cells, phagocytosis process. Next is we're going to talk about the molecule, movement of molecule. As I mentioned earlier, the molecules can also move through the protein channels. Those protein channels are called as porins. With the help of this porine, the molecules can move inside and goes outside. There are aquaporins, which are seen in bacteria, plants, and animals. There is a channel that is present in the plasma membrane to transport the water molecules. That is called as aquaporins. Next is the movement of molecules. If only one type of molecules move across the membrane, that is called uniport. Synport, two different molecules moving in the same direction. Just observe the diagram, children. If only one molecules move inside at a time across the cell membrane, that is called as uniport. This is through a protein channel. Synport, two different molecules move in the same direction with the help of the integral protein, same direction. Antiport, two different molecules moves in opposite direction. That is called antiport. Next, we are going to talk about cell wall. The next topic is about cell wall. Okay. So now, peptide transport also requires ATP. Yes, the doubt by Madev. Peptide transport requires very minimal amount of energy. That energy, it is used by the carrier molecule. It is used by the carrier molecule. It is not used by the substance. Peptide transport requires less energy because the peptide transportation, it occurs mainly with the help of the carrier protein or integral protein. You got it? Yes. The next topic is about the cell wall. Cell wall. We're going to talk about the cell wall. Children, I'll give you 5 minutes time. Just copy this slide. Layers of cell wall. What it contains, all those informations are there. Just do that. After 5 minutes, I'll explain the cell wall. The cell wall is there. And then we're going to talk about what are the components of the cell wall, cytoplasm, what it contains, cell organelles, all those things I'm going to discuss to you. So now, just write the content alone here. If possible, you can draw the diagram also. I'll give you 5 minutes time. I want you to write the information and then upload it in the WhatsApp. Okay, children, do it now. Do it fast. Okay, children, welcome back. And few of you, Madesh, Balakrishna, and Kishore, we have sent it the documents. I have seen that. Others send it fast. We'll move on to the next topic. The next topic is about cell wall. As I mentioned earlier, there is one more protective layer that is present in plants. This layer, it is actually partially it is living and non-living structures. This is present exclusively in the cell wall. That is the reason why all the plant cells are thick, hard, rigid, and these structures are secreted by the components from the cytoplasm or from the protoplasm. It is a supportive layer and protective layer in plants. What are the different layers are there? The layers of the cell wall, it includes the middle lamellae, primary cell wall, secondary cell wall. You can see that the middle lamellae and primary cell wall and the secondary cell walls. This structure, the typical structure, you can see that the middle lamellae, it contains cementing layer, attaching layer between the primary cell wall of adjacent cells. Middle lamellae, which connects, this is one cell, this is another cell, that two cells are cemented, joined by middle lamellae. Got it? Cementing layer. That cementing layer, it is present between the primary walls of adjacent cells. That is the reason why it is called middle lamellae. It is present between the primary walls of two cells. And that middle lamellae, it consists of pectin, calcium, and magnesium. Primary cell wall, that is the one that surrounds the plasma membrane. And that primary cell wall only has the plasmodesmata. Can anyone tell what is plasmodesmata? Do you remember that what is plasmodesmata? What is plasmodesmata? Can anyone tell? You have learned that in transportation in plants. Do you remember, children? Charvi, can you tell that? Deepak? Kadiravan? Kishore is there? Balakrishnan. Can anyone tell what is plasmodesmata? You have learned that. Do you remember, the children, what is plasmodesmata? Are you all able to hear me? Are you all able to hear me, children? Do you remember that when I explained about transportation in plants, there are three different types of transportation takes place. Can you recollect that? These are all channels that are present between the endodermis, cortex, epidermis, cortex, endodermis. Can you recollect and tell what are the three modes of route of transportation? Three routes of transportation, children. Do you remember that? In transportation in plants, do you remember that three modes of transportation? What are the three modes of transportation? Apoplast, symplast, transmembrane route. Apoplast, symplast, transmembrane route. Do you all remember that? Shall I show the picture, children? Do you remember that, children? Yes. In that, symplast transportation, it occurs through cytoplasm. Do you remember that? There only the plasmodesmata is present. This plasmodesmata, it is formed by the primary cell wall, cellulose, hemicellulose, and pectin. Secondary cell wall or secondary wall, it is inner to the primary wall, and that itself it has three layers: outer, middle, and inner layers. These are the layers of cell walls. Next is the molecular organization of cell wall, what it is made up of. The cell wall, it is made up of matrix and macrofibrils. These macrofibrils, they're all made up of glucose. Glucose molecule, the glucose molecule, 3000, 6000 glucose molecule, they combine together and they form a cellulose chain. Hundreds of cellulose chains, they form the elementary fibril, that's called micelle. And those elementary fibril, it forms microfibril. Microfibrils forms macrofibrils. The cell wall, it contains polysaccharides, pectins, gums, tannins, silica, and wax. These are all the molecular organization of cell wall. Now you know that how the cell walls are formed. Cell walls are formed by macrofibrils. Macrofibrils are formed from microfibrils. Microfibrils are formed from elementary fibril. Elementary fibrils are formed from cellulose chain. Cellulose chains are formed from the glucose. So basically, all these cell wall are mainly the carbohydrate molecule, glucose molecule. Next is the components of cell wall and their function. Children, please make a note. Just make a note. I'll give you time for you to make a note. Just take notes of these components, what is listed here, all those things, and then take a photocopy and send it to me. I'll give you 5 minutes time. See the tannins, and these are all the waste substances. Gum, tannins, these are all waste substances produced by the cells. Tannins. These are the components of cell wall and their function. Please make a note, children. I'll give you 5 minutes time. Welcome back, children. The components of cell wall and their functions. Mainly, these are the components that are present in the cell wall: cellulose, hemicellulose, lignin, pectin, cutin, and suberin. The cellulose, you know that how cellulose are formed, right? The glucose molecule combine and forms the cellulose. There are the structural component of primary and secondary cell walls. Hemicellulose, it consists of arabinose, xylose, mannose, and galactose. All of these are also the carbohydrate molecule. They binds the cellulose fibers. It helps in binding of glucose molecule. Lignin, coniferal alcohol. It provides stiffness and rigidity. Next is pectin. It acts as a pillar substrate in between the components. It is made up of glucuronic acid and galactonic acid. Next is cutin, made up of fatty acids. It prevents loss of water. Suberin, again it is made up of fatty acid. It is impermeability, prevent infections. It mainly prevents the infection and also it prevents the entry of water molecules. That is about the components of cell wall. Next is cytoplasm. This topic from cytoplasm, all the other cellular organelles, all those things we'll discuss in the next class, children. And now if you have any doubt, I'll just clarify your doubt for another 5 to 10 minutes. Next topic, that is cytoplasm. We'll see it in next class. I'll just inform you when we have next class. Probably if Sunday, if you are free, we can have a class on Sunday also. Sunday, you just tell me we can have a class on Sunday. We'll have it. Okay, children. The next topic we'll discuss in the next class. If you have doubt, I'll clarify your doubt. Thank you very much, children.